Assalamu alaikum and have a happy good morning. Welcome to my YouTube channel. Student, today we are going to talk about our next lecture about the organelles of the cells. And this we are going to talk about the lysosome structure and its function. Student, lysosome work actually as a eater. Like in our daily life, someone eat a lot of things, so we can collect. Oh, you are eating so much thing. So actually, lysosome is actually working like a big eater in our daily life, like in our home. Uh, my brother is eating more and more. So, and the cell, the lysosome also work as eater. They eats all the content present in the cell which are expired. The word lysosome means splitting, breakdowning. So the thing which are present in the cell, so it break down, and so mean body. So it is the body of breakdowning and splitting, and then eat this and digest this, and then send out these from the cell. Like in our home, if someone is if my brother is eating more and more, if he find anything in whom, so he collect these and eat all these, and something which are not used in our home, so they collect all these and work as a rubbisher and send this outer side, and also eat the part of other. Like someone have brought something from the bazaar, so they also eat the part of other person, like my mother, like my sister, so they also eat the part of other. Actually the lysosome do the same thing is like in our home. So the home is like the cell, so in the cell it also work as a collector and then eater and then digester, and then it collect them and send them outside. So they also work as a collector and also a rubbisher. Okay, so this was a short introduction to the lysosome. There is a single membrane bounded organelles. Okay, student, this is a single membrane bounded organelles. So student, in the cell there are some organelles which have double membrane, like nucleus, mitochondria, Golgi apparatus, and some other content have double membrane, but the lysosome have single membrane bound organelles. Okay, next, the word lysosome means splitting and so mean body. So actually at work is a splitter of the food and then excrete them. Okay, it is also known as a rubbish collector. So I mentioned that it collect all the expired thing from all over the cell and then digest these and convert them into digestible things. Okay, they were discovered by Didiu in 1949. By Didiu, a British scientist in 1949. Okay, formation. How are they formed? This is very important. Talk about the lysosome. Actually, I have mentioned this diagram here. You can see here. This is the diagram of lysosome. Why this is the diagram of a cell? So in the cell, this part is the nucleus. You can see here, this is the nucleus. Why this is the endoplasmic reticulum. You can see here en reticulum. You can see here, this is the endoplasmic reticulum. This is nucleus. Okay, and this portion is the Golgi. Okay, Golgi. This is Golgi. Okay. So the actually lysosome is formed from the degrading of lipid, protein and carbohydrate in ER. Okay, so student, ribosome are actually found in the endoplasmic reticulum. They are actually formed in the nucleolus, which is found inside the nucleus. It's called nucleolus. There is no flesh. Okay, this is called nucleolus. This is nucleus, but it is nucleolus. So in the nucleolus, the ribosome is formed, not lysosome. It is ribosome. I'm talking about ribosome. So the ribosome is formed. It come out of the nucleus and then attached with the endoplasmic reticulum. So now there, the cell oxidize the food by the oxygen, like we are saying in the cell respiration, O2 plus C6H12O6. So on this way ATP is formed. In this way protein, lipid and carbohydrate are also synthesized. So these protein, carbohydrate and the lipid are present in the endoplasmic reticulum. When they are moved to the next part, the Golgi apparatus. So endoplasmic reticulum is of two types. One is the smooth endoplasmic reticulum. So I can write as a smooth ER and RER. Okay. One is the SER, smooth endoplasmic reticulum. Second is the rough endoplasmic reticulum. So when these particles are present in the endoplasmic reticulum, so they are sent to the next part, the Golgi apparatus, through the in the form of small vessel-like structure by the smooth endoplasmic reticulum. So you can see here, this part is called the smooth endoplasmic reticulum. From there, the lipid, protein, and carbohydrate are sent to the next part, Golgi apparatus, in the form like these vessels. Okay, these vessels carry the lipid, protein and carbohydrate to the next part, the Golgi apparatus, like in this part. So when they reach the Golgi apparatus, so Golgi apparatus is actually work as a machine which convert the product produced by the endoplasmic reticulum into a form which is useful for the working for the activities of the cell. Okay, so when they are when they pass through the Golgi apparatus, so some of the lipid, protein and carbohydrate are converted into energy form, while when there is formation of the lysosome, so they are converted, so they are modified in these membranes into another form which we call as a lysosome. So they come out as a like this structure. Okay, you can see here, this is structure of lysosome. So actually the conclusion is this, that the endoplasmic reticulum and particularly the smooth endoplasmic reticulum send the lipid, protein and carbohydrate in the form of vessels to the next part, the Golgi apparatus, where it is modified and converted into a form called as a lysosome. In the form, and they are passed from the Golgi apparatus in the form of vessels which we call as a primary lysosome. It is called primary lysosome, and then they stay in the cytoplasm. You can see here, this whole is cytoplasm other than the organelles. So then they started their working. So let's come about structure. You can see here, student, there are two things, the red cells and the green cells. We call these as also as hydrolysis, which are the combination of protein and enzyme, and I could also say hydrolytic acid and phosphatase enzyme. Okay, so actually if we talk about the function of the lysosome, let's come about its function, how it performs its function. This is the function we are saying about the function of the lysosome. So if we are saying about the function, so in the function we found there two types of enzyme. One is the hydrolytic acid, and second is phosphatase enzyme, which are protein and other enzymes. Actually, the hydrolytic acid is a type of acid which have a specific pH. Due to its pH, it converts the expired material into a digestible form. And another work is when any parian content enter to the cell, like if we say now is the coronavirus. So when the coronavirus enter to our cell, so in our cell the lysosome work is a eater. It envelop the coronavirus. Like I'm showing you diagram is like there. This is our lysosome. So this is coronavirus entering our, okay, this is coronavirus style. You can see here this is the lipstick of the coronavirus. It is not actually, but I am only showing here. Okay, actually when the coronavirus enter, so it start fighting. The lysosome start fighting with the coronavirus, which I mentioned coronavirus in our now time. Okay, so when it enter to the cell, so the lysosome directly start fighting with it and directly envelop it, like is there. Like this is the I'm making again this, like it directly chew the coronavirus. It envelop directly the coronavirus. So then the coronavirus cannot perform its function in the cell, so directly enveloped by the lysosome, because due to the presence of some enzyme, some acid, which we call hydrolytic acid, which have a specific pH, and phosphatase enzyme. Due to these, it enveloped directly the coronavirus. Okay, and performing, normalizing, staying its common normal pH. This is the cell. So every time we need for the acid the H positive ion. We need H positive ion to maintain the acid its function. So every time the cell provide the lysosome H positive ion in order to maintain its specific pH. Okay, so its pH is maintained by the cell by pumping every time the H positive ion. So I said that the function of the lysosome is enveloping the expired material. Another function is this, like if it is this is mitochondria which is expired. So the expired mitochondria is also enveloped by the lysosome. So directly, like I am making again this like this is the what lysosome. So and this is the expired mitochondria. So in the cell, when the mitochondria expire, so it also eat this directly. It's like this. I'm making this is like this, directly eat. If other component like the Golgi apparatus, other component are expired in the cell, so directly it envelop it. And what are they doing after enveloping? They send directly into vacuole. So there are small vacuole when they send it to this, and then in the palm of some pipe-like structure which come out sometime from our face. So they come out in this palm, and because they are expired, okay, so they come out of the cell is expiring. Okay, Next, we will talk about if we talk about its disease. So mainly we found a disease which name is glycogenesis type 2. Okay, glycogenesis type 2. It is a disease because we say that in previous video I have mentioned about the structure of liver and its function. You can see my video about the liver. The glycogenesis is a process in which the excess amount of glucose are converted into the glycogen, which are then stored in the liver and also other tissues and muscles. Okay, so in the glycogenesis type 2 disease, when the amount of glycogen are increased in our liver tissues and our muscle tissues, because there is an enzyme which we call as a glycogen, insulin, or the pancreas, insulin, which increase the converting of glycogen into glucose. So when this enzyme is not present, as is absent, like other enzyme also work like this, but when it is absent, so the excess of glycogen are not converting into glucose, and there is need in the body of glucose because body need more metabolic activities, so there is a need of more energy, and the glycogen amount is also increased in our liver and also the muscle tissues. So this disease is called the glycogenesis type 2, and this disease enzyme is absent due to which the glycogen is amount is higher in the liver and also the muscle tissues. So in this disease, the lysosome also stop working because there is high amount of glycogen stored in the cell. So the lysosome also cannot perform its function properly because it cannot receive its common food in order to normalize its function, in order to remove the toxic material from the cell. So in this disease, it also stop working. If we say about another disease which we call as Tay-Sachs disease, name is Tay-Sachs disease. Okay, this name is Tay-Sachs disease. Pronounce properly. Okay, Tay-Sachs disease. Okay, Tay-Sachs disease. Are you know now? Okay, in this disease, when the catabolic activities or the catabolism of the lipid increase in the tissues. What is catabolic? Catabolism is a process in which the higher, like the complex thing convert into simpler. Okay, the big thing convert into small. Okay, so when the catabolic activities or the catabolism of the lipid increase in the tissue and the cells, so it leads this disease because the number or the amount of the lipid increase in our tissues and our cells. So we don't need more amount of lipid. So in this situation, our lysosome also stop working because the lipid amount is so high. Our lysosome cannot bear to remove these out from the cell. And when this amount or this disease occur in our brain, so it causes our brain disorder, which we call retardation of brain. Retardation of brain, in which the lipid amount and our blood vessels which are present in the brain. So when the vessels in the brain stop working, so we will not respond to any change. And if the brain stop working, so brain is our like in a school as a principal. So if there is no principal, the teacher cannot work. In this way we call as a retardation of brain, and the person is actually we call it it is non-aware, it is unconscious of everything. So student, this was about the lysosome structure and function. If you have any doubt in its structure and function and other activities, so you can mention me in comments, and please subscribe my video to receive more of my lectures.